In our group, we tend to focus on oncogenes. So these are genes that are normally characterized as being very important in cancer. And these are faulty genes, genes that send a relentless signal back to cancer cells to tell them to proliferate. And what we can do is we can delete these genes from our cells, we can put them back into the cells. What we can also do is then we can see if there's some different way for us to manipulate it and see if we can switch them off. By doing so, we can see what other components are important for tumor progression. We can have a look at these cells and see whether they're important in terms of tumor invasion, in terms of tumor progression and tumor metastasis. When we have found something interesting in the lab and something that we think that is important in terms of cancer progression, we can really use our collections and our connection with the research nurses, and we can utilize our patient samples to really link what we do in the lab and really connect the breast cancer cells, the models that we use in the lab, with those of patients. And by doing so, we can collate all this data together and use it to predict which patients will do well on therapy, which patients might relapse on therapy, and to help us better understand how we can actually overcome and help the significant portion of patients that don't ultimately benefit. The ultimate goal for us would be to find a biomarker that we could potentially test out in a standard blood test, which will be able to tell you whether the patient can respond to the therapy. If they're a responder, great, give them the therapy. But if this biomarker is high in the blood, we should be able to say this patient might actually relapse on therapy after a number of years. And this is ultimately leading to a more personalized approach to medicine and not doing a one-for-all treatment.